In this video, we will discuss about the in vivo results of GlyTR1 and GlyTR2 on mice model. So one of the mice that they have selected for their studies is humanized mice, and the reason why they selected the humanized mice because these the antibodies that they have used in the bite, that is against human CD3, does not bind to the mouse CD3. That's why they have used humanized mouse for their in vivo studies. Now let us understand the results that they have presented in this figure. So how they did their studies? They distinguished between tumor cells and the GlyTR1 and GlyTR2 through different signals. That is, tumors are conjugated with this luciferase signals, whereas the GlyTR1 and GlyTR2 are conjugated with the fluorescence signals. So panel A shows the difference in luciferase and GlyTR1 signals, and panel 2 shows the luciferase and GlyTR2 fluorescence signals in TNBC. Both of them are in TNBC with this mice MHC-1 deficient. At the same time, this COSMC gene knockout also, because it is essential for the high TN expression. It is not essential for this GlyTR1. Still, they have used the same cell lines in both the conditions, and it will not affect the results for the panel A. Afterwards, they did their GlyTR1 studies on CAPAN-1 cells with MHC-1 deficient, and also on these TNBC cells with MHC-1 deficient, that is shown here. In this case, the COSMC gene knockout is not required, as this GlyTR1 is specific to the beta-1/6 branching, which is expressed in both the cancer cell lines. But this MHC-1 deficient is required to prevent the cytotoxicity because of the CD8 plus T cells. So as we can see, compared to the control for this this CAPAN-1 MHC-1 deficient cells shows tumor suppressions with time, and similarly for this TNBC also they are able to see tumor suppressions with times. And when these things are quantified in the, for example, for this CAPAN-1 MHC-1 deficient cells, they are able to see the tumor suppressions is significantly they are able to see tumor suppression is significantly high compared to the control case, where the tumor rises continuously with time. And similar results they have seen for the TNBC also, that the tumor suppression is significantly higher compared to the control case. Afterwards, they did the similar studies for the GlyTR2 bite antigen. So in this case, they use this TNBC, that is MDA-MB231F cells with MHC-1 deficient, and also the COSMC gene deficient, because the COSMC gene deficient will cause the high expression of this TN antigen. And similarly for this SKOV3F also, that is ovarian cancer cell lines, they use MHC-1 deficient. However, they inherently have this TN antigen, so they don't need to do further modifications in this ovarian cancer cell lines. And in the case of this TNBC with MHC-1 deficient and the COSMC gene knockout, they are able to see tumor suppressions, but they can see that tumor relapse again started to occur in the later days. But still, the tumor size in the later days are significantly lower compared to the case where there is no bite antigen is used. And these quantifications are shown in this panel H, where we can see that the tumor size even after 35 days is statistically significantly lower compared to the control case, and also compared to the case where they use less amount of GlyTR. But even in the less amount of GlyTR, the tumor suppressions is significantly lower. Tumor suppression is significantly higher compared to the case of the control, as the p-value is less than 0.05. And when they check it for these ovarian cancer cells, they are able to see there is no difference. They are able to see there is a tumor suppressions with the increase in time. And they did this. They did check this ovarian cancer cells also for the GlyTR1, and for that also they are able to see tumor suppressions consistent with time. And these things they have quantified in the panel I also. And here we can see that the tumor suppression is significantly higher for both this GlyTR1 and GlyTR2 compared to the control case. So this indicates that both the GlyTR1 and GlyTR2 are efficient or provide acceptable results even in the in vivo studies. So that's all for this video. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. So thank you for your time.